to today's speaker, Dr. Kelli Hoover from Michigan State University, who's an expert in the insect cell microbe interaction and invasive species. Let's give her a warm welcome as she shares a story about spotted lanternfly.
Well, thank you for inviting me. This is a real pleasure, not only to get to hang out with Brian for some days, who I've known since grad school, but it's also an opportunity to come here. I never had been to before. So I've been working on invasive species most of my career, and I got involved in spotted lanternfly research in 2019 when it had already been in the US for a couple of years. I couldn't get away from it because the dean was calling on, all hands on deck. So I got involved, and I'm going to talk to you about ecological interactions between the insect's host plants and the insect and how they impact their feeding hosts. And also I'll talk to you about how we've been looking into natural predation in nature, as well as possibility of introducing some biological controls. So I'm going to start with where is the insect found now? It's now in 19 states. Pennsylvania right up here is almost entirely covered. The only part of the state that doesn't have infestations is in the very far north. And Erie County is up there in the corner by Lake Erie, which is the grape growing region of Pennsylvania. So you can imagine that people are pretty nervous about it. And unfortunately, it's also up in the Finger Lakes. So it is affecting vineyards there. So the latest infestation was found in South Carolina. It's not on the map, but that was just discovered very recently. So it's getting closer and closer to you. And wherever you have tree of heaven growing, which is most preferred host, it can show up. Now, it doesn't require tree of heaven, but fitness is greatly reduced. They don't have access to it. So here are the life stages, and up in the top left corner are the egg masses. This is an overwintered one that is starting to crack. So the eggs are probably getting ready to hatch. The first three instars are black with white spots. The fourth instar has aposematic coloration. So remember that. And the adults look really different from the nymphs and have red in their hind wings. And they flash those as a startle display when the predator tries to come for them. They're easy to sex. The female has this red spot on the tip of her abdomen, the males don't have it. And notice all the yellow on the abdomen. As they feed for about a month before they develop an egg load, those yellow sides start to pop out on the corin and also the abdomen. And it also happens with the males as they're developing an egg load. So you can tell whether they're gravid or not. And here's a female laying eggs, and they lay eggs on every kind of surface you possibly imagine, including inanimate objects, non-hosts like conifers, but they never feed on conifers. Why they do it, I don't know, but you can see what she's doing is she's laying wax down over her eggs. They're laid in the fall, so they're laying eggs now in Pennsylvania. And the eggs overwinter and start hatching in the spring. There are about 35 to 40 eggs in a given mass. And females, as I'm showing here, do protect their eggs with a waxy covering. As a consequence of that, you almost never see eggs preyed on. The predators that are out there are just not touching them. On occasional slug, we've seen eat some, but other than that. And as I said, they'll lay eggs on everything. So one of the reasons they're highly successful is they're excellent hitchhikers. So they can make big leaps. That's how they've gotten as far as they have. Because they'll lay eggs on yard furniture, tires, underwells of trucks. They'll hop onto railroad cars. They've been found hitching rides in the cargo hold of airplanes. So to California, which is very scary for them because of their wine growth. So they're very successful in part because they have this broad host range. They can feed on over 100 different plants, although not all of these are in the US. They select hosts based on the proximity, what's around me that's suitable, and seasonality. because as they go through each life stage, they get more picky about what they're eating. So the host range narrows, particularly in the fourth instar and adult. And they do have a few favorite hosts. The nymphs will feed on roses, cultivated rose as well as multiflora rose, and on grapes, wild and planted grapes, table grapes and wine grapes. Tree of Heaven is their most preferred host. They co-evolved with it in China, and it was introduced in the US, as some of you may know, in the late 1700s. So it's been around a long time, and it's really hard to get rid of because you have to kill the roosters to get rid of it. And the adults prefer Tree of Heaven, and they'll also feed heavily on grape. And as Tree of Heaven goes into senescence, it's an early senescer. They move on to red and silver maple, although we have way more red maple in Pennsylvania than we do silver. And other trees like sycamores, river birch, willows, etc. So this is how bad it can be. And this may look bad, but seeing it in a picture is nothing like seeing it up close and personal. You can walk up to a tree and just go like this and knock thousands of them to the ground. And they do attack grapes in very large numbers, and that is our biggest economic impact is on grapes. They do feed on fruit trees, but we have not found them to cause any damage. They only stuck around when they started hitting apples for a couple of weeks, and then they flew off. And the growers didn't notice any fruit yield or anything. And they're a really big pest on fruit trees in South Korea, where they were introduced, but they're not here in the US. We don't really know why. Okay, so when the populations are really large, this is what it's like working in the forest. You think that's rain? It's not rain. It's honeydew coming down in huge amounts, and it attracts stinging insects and ants. And also they cause weeping wounds on trees. It's amazing how they will push their stylids into a tree through the bark, and they end up making a hole about that big. And so other lanternflies start congregating, trying to get to that hole. So whatever they're doing, they're probably affecting source sink dynamics of the sugars in the tree. And I'll talk more about that later. Interestingly, honeybees collect honey and make honey out of it in the fall. So September honey tends to be dark in color because they fed on Tree of Heaven, which contains some toxic chemicals. But some people, they're not toxic to us. Some people really like that. Some beekeepers sell it as a delicacy. I think it tastes awful. I taste it and like, no, thank you. All right, so this is what happens when all that honeydew gets produced. We get sooty mold, and it's amazing how many trees in a heavily infested area have a completely covered trunk with black sooty mold that grows on honeydew. And obviously, if it hits leaves, it's blocking photosynthesis, so it's not good for the understory either. So we decided to start looking into, we were getting asked lots of questions about, should I be worried about this insect when it's hitting my trees? Especially if I'm in a production nursery and I'm trying to grow trees to size to be able to sell. So we decided to look into this in a couple of ways. We did studies short-term in a common garden that I established on 2 acres in southeastern Pennsylvania. This was before it was found in my county. So we were having to drive 3 hours each way to do research. We planted silver maple, red maple, black walnut, and Tree of Heaven, all our preferred hosts for this insect. And then I set up some long-term studies that ran for 4 years on using multi-tree enclosures that were planted with 3 different species. And I'll talk more about that in a minute. We tested all life stages in the long-term studies for 4 years, and 4th instars in adults only in our short-term 10-day studies, because we wanted to see on these young saplings, because they were small trees at the time we were doing this. We were pretty sure it wouldn't take very long to see effects. And we collected data on gas exchange, non-structural carbohydrates, essential nutrients, and diameter growth. So the first study, this is the cone garden, and these are enclosed in these huge bags that are being held up at the top by this metal post that we put in the ground, because when thunderstorms come through, they can knock these things down. You don't have them staked up. And the work I'm going to talk about from this has been published, and we used 4 different densities of instar lantern bodies, as you can see there, and we tested the fourths, silver maple and black walnut. Black walnut is a strong preference for instars, and silver maple and tree academy results. And this is what we saw with soluble sugars. We didn't see big impacts in leaves, but on the branches, there was a significant decline as you went up in density, although they weren't always significantly different. But it is sort of a dose-dependent response of reduction in soluble sugars in the branches, which is where they're feeding. So they're not feeding on leaves, they're feeding on branches. First instars can feed on the veins of leaves, sometimes they'll do that. Then the seconds and thirds are feeding on softer branches. Fourths have moved on to feeding on woody stems, and the adults feed on trunks and very, very large branches. So diameter growth was impacted the following season. We saw about a little more than a 50% reduction in diameter growth the following spring of these trees. Then this is our multi-tree enclosures. We set this up in 2019, and we planted silver maple and willow weeping willow in each one of these enclosures. There were 10 of them. And then half had tree of heaven and half had river birch instead. This initially was set up to test the hypothesis that lanternflies could develop and reproduce without ever having access to tree of heaven. There was an assumption from both APHIS and many state departments of ag that they could not survive without tree of heaven. So we decided to test that hypothesis. And the first year we released a lot of first instars into each cage. And in the second year and subsequent years, we used fewer, and we started with third instars because we were getting a lot of mortality in the first couple of instars, and we had already shown in that first year that we could take them all the way through to adults. So in the second year, we got a little bit more land. This was on an extension educator's property in Southeast PA. We set up 2 control plots with trees that were enclosed but never exposed to landflies. And this is what we saw for gas exchange on the y-axis. So carbon assimilation, stomatal conductance, and transpiration were all higher in the treated trees. So we were seeing compensatory photosynthesis, which we didn't know which way it would go. It could go either. This isn't unusual for plants to try to make up the difference of all the sugars they're losing by increasing their rate of photosynthesis. But once you get past the second year, this wasn't happening anymore. We did see photosynthesis decline. And non-structural carbohydrates were also reduced. They were higher in the control than in the 2020 treatment or the 2019 treatment. And total non-structural carbohydrates sort of have a similar pattern. And when you start losing starch, which is stored in the roots, and it's kind of the bank to get through the winter, trees start having trouble. And that's what's killing grapevines, is they lose their root storage of carbohydrates, and they can't get through the winter without mortality. We also saw a reduction in some essential nutrients that are important for photosynthesis compared to controls. And in this study, what you're looking at is the annual diameter growth on that y-axis, and on the right y-axis, this is the average insect pressure per cage. And the dashed line is the insect pressure. And so what you're seeing here is that in the second year of the study, the amount of growth declined. This is across all trees. And in 2021, we reduced the number of insects that we wanted to see if the trees could recover. And so we reduced the population that year, and sure enough, they did recover. To some extent, not fully, they did recover some. And then in 2022, we increased the pressure again, and again, we saw a decline in diameter growth. So these trees are now completely open. They don't have cages on them anymore, and they're actually too big. This is something really interesting we saw at the Common Garden. Somebody had mentioned peeling back a grapevine branch, and that where the lanternflies were feeding, they were seeing these dark dots. So at our Common Garden, we peeled back the bark where they were feeding, and this is what we were seeing. But notice, tree of heaven is not responding. The plant that they co-evolved with. So I don't know if there are elicitors that lanternfly is producing that are not triggering a response here. That's still being investigated, and we don't know if this is just necrosis or if it's an induced response of callus formation, which could be what's happening because it's been shown to occur in the brown plant hopper, which is a pest on rice. They produce hamps that induce these kinds of responses. So in summary, the effects on trees are that lanternfly is really a plant stressor, and it can manipulate source sink dynamics and reduce starch concentrations in the roots co-occurred with the decline in diameter growth. When feeding pressure was reduced, trees could recover, but we were finding that the smaller, younger plants were more effective than the established, which makes sense. And they are killing large tree of heaven in the field. There's a lot of dead stems, and some of them are right next to freeways, and that makes us nervous. They might fall on somebody. But these insects move all the time. What we think they're doing is they're exhausting the food source, and then they fly off and go somewhere else to find better food. They're not in grapes the whole summer. They may be on there as early instars and they leave. We don't know where they're going or if they're going to come back, but most of the time they do as adults. So the negative impacts that I showed in the 4-year study is probably worst case scenario because they had pressure for 4 years straight. And in nature, as they would probably have moved. In fact, they were flying to the edges of the cage trying to get out. So economically, the greatest cost to the forest products and ornamental industries is dealing with a quarantine. You have to get a permit to transport anything out of quarantine. And you have to make sure you're not transporting any life stages. And we have had nurseries that have had infested egg masses on plants that they've received, and they had to be tracked out and collected, destroyed. The impact on grapes has been really dramatic. The big notice here is they've had to increase their application of insecticides from 4 to 14 per season. They're spraying mostly by fenthrin to kill the adults. And this has increased the cost for insecticide use from $54 to $147. So growers are really suffering. And as you can see from some of this up here, many of them are losing some of their vines. They've had winter mortality. Sometimes an entire planting is killed. So it's a really serious problem on grapes. And this is what a dead vineyard looks like. So this was in the summer. So what are we going to do about this? Are there controls out there besides having to spray tons of insecticide? A lot of the treatments being done with a neonic called dimetethron. Imidacloprid doesn't work very well on spotted lanternfly. But if they're looking at contact kill, they'll use a pyrite for it, like fenthrin. But there are natural enemies that are being investigated. There is a classical biocontrol program that's going on with USDA on Dorianus sinicus. This is an ectoparasitoid, that you can see right here. and it attacks the second and third instar nymphs. They're working on host range testing right now, and they're also exploring how to mass rear it. Because you can only rear it in the insect. And the problem is that the insect is very hard to rear in captivity, which is why I was using planted trees. So it's gonna be tough, but I think they may be able to do it. So there's also an entomopathogenic fungus that causes localized natural epizootics. And this is what you're seeing here. This is a lanternfly killed by Bavaria bassiana. It's being investigated. Ann Hayek worked on it at Cornell for a while. Stefan Jaronsky is working on it now. But it doesn't work very well in the field. And I think the reason is that you have to get really good coverage. And because they jump off or fly away as soon as you start spraying something, they're not getting good coverage. So I'm not sure if they're going to be able to use this. So we looked into natural control. There is egg parasitism in the field, but it's very, very low. But there are a bunch of endemic generalist predators that are feeding on these insects. So we set up a Facebook page. Ann Johnson is the one who did all this work. She set up a page called Birds Fighting Bad Bugs. And she set it up in the summer of 2020 when COVID was shutting everything down and everybody and his brother were becoming bird watchers. So there we got almost 1,300 unique reports over a couple of years. And the work from this has been published, but this is what it looked like. So the biggest number of predators occurred in the arthropods. So we had wheel bugs, spiders, all kinds of things that were attacking them. And that was followed by birds and even in the occasional mammal. So we are seeing this go on in nature. But we also know that lanternflies sequester toxins from triacetamine. What they're sequestering, in addition to alkaloids, are plazmids. These are bitter tasting compounds. They taste bitter to us. It's that icky smell from tree of heaven. If you cut into a branch, break a leaf, and smell it. It smells terrible. But these are structurally complex and highly oxygenated degraded terpenes. And there are several different ones. And we have a lanthone as a standard, as well as 5 other standards from a German natural products chemist who isolated these compounds from tree of heaven. So we were able to explore concentrations in the insects and in the plant. So they were in the phloem. We had to make sure they were in what they were feeding on. The concentrations were highest in the adults and in the egg masses. And I come back to egg masses are not being preyed on. There is some carryover of these compounds into the first and second instars. And then it starts declining until the lanternflies become adults and then the concentrations. In the tissues, we found concentrations highest in the gut and salivary glands, followed by the ovaries, which is probably how they're getting into the eggs, and the cuticle. And long story short, a paper that just came out a few months ago, arthropods did not show any evidence of difference for insects, lanternflies that had fed on tree of heaven versus those that never had. So we rear them through without access to tree of heaven on mostly grain. And they do pretty well. So here's the different sources to give you an idea. Concentration is on the y-axis there. And you can see that the lanthone in tree of heaven is not as high as it is in the insect. If they're field collected, that indicates they're concentrating, they're sequestering. And this is the other plazmoids. But the field collected lanternflies have significantly more than when we reared them in the greenhouse on potted tree of heaven. They really go for vigorous, big plants because they get more phloem out of them per minute. The little plants, they're just not acquiring as much stuff, so it is harder to rear them in captivity. And we did see that these levels were lower, so we were careful about which insects we used in subsequent studies. So we did a study on using nest boxes to look at bird predation. So these were house wren nest boxes at the Arboretum, and placed 10 mealworms to get the wiggle, so they would catch the bird's attention. And 10 lanternflies into each cup, and they were individually marked whether they had the lanternflies had fed on tree pennon or not. So they were frozen and then thawed for this experiment. Because we didn't have them in our county yet when we did this study, and so they had to be dead. So we observed to see what the birds would do with these insects, and we set up trail cams as well. And checked the cups at 24 hours to see how many were missing by source, and then continued to check for a little bit longer. And this is in press, by the way, in Journal of Chemical Ecology too. Anyway, so here's the proportion eaten of the lanternflies on the y-axis. And across all life stages of the nymphs, they ate significantly more that had not fed on tree pennon. So birds seem to be able to tell the difference. They can detect the chemical and avoid it a little bit. Well, we couldn't do adults though, because the lanternfly adults were not present when the birds had stopped nesting. So we made suet cakes, and we had two-sided suet feeders. One side had lanternflies crushed up in it that had not fed on tree pennon. The other half was lanternflies that were fed on tree pennon. We had 3 field sites. One was at the Arboretum. I was telling Franny about the Arboretum. And 2 feeders at each site, and they all had trail cameras. So you can imagine how much video over 2 weeks had to be viewed. 2 weeks of video at 6 locations. So she counted the number of pecks and then did an analysis and found that birds pecked more suet that contained lanternflies that had not fed on tree pennon. And she also did there's other graphs in the paper showing different bird species, chickadees versus, woodpeckers and so on. So they can tell the difference. And interestingly, there were mammals. So we sometimes had raccoons, mice, squirrels, and opossums. And the opossums and the raccoons never fed on the tree pennon spored chemical lanternflies. They only fed on the one that had not had access. And the mice and squirrels, out of 300 of them visits, 3 times they touched the other suet, and that was it. So for birds, here's our nesting house friends. They are affected by sequestered defenses. They appear to avoid, this was the interesting part, they never fed their chicks a lanternfly that had fed on tree pennon. They avoided feeding them to their children. They liked them themselves, but they couldn't give them to their offspring. So another thing switching gears, we started to look into, after having seen all of this predation going on by arthropods out in nature, we decided to see how effective they are at reducing numbers in an experiment. So we tested green lacewings, convergent lady beetles, multicolored Asian lady beetles, and egg masses were used, as well as first and second instars from these smaller predators. We had cages with 1 grape and 1 tree pennon plant. We grew in 25 nymphs, each life stage tested separately, and 10 egg masses or 10 adults. And the number in parentheses is how many predators were there compared to prey. Spine soldier bugs, is anybody familiar with that insect? It's a predatory stink bug. Yeah. And it turned out to be the winner. We did 3 reps of each predator at each life stage and controls, and then we monitored numbers for a week. All of life stages look like this. So I'm just going to show you a couple. This is the 4th instars. In 1 week, this is the control up here. So this is natural mortality. Wheel bugs produced this much mortality, and it wasn't as high as we expected, but I think it's because they were eating each other. We had 8 of them in there, and there was a lot of chemicals going on, so we've learned that we can't do that. In nature, they don't, they're not together, get a crap. And here is our spine soldier. Podesis maculoventrus. It's really good at knocking down numbers, and the mantises did a pretty good job too. And this was adults. So both species of mantis plus spine soldier bugs were by day 3 had pretty much eliminated most of the lanternflies, and by day 5 they were all gone in the Carolina mantis experiment. There's been a paper that has come out since this work was done, showing that there is that the mantises have a functional response to lanternfly populations. So eggs were well protected, but mantis and spine soldier bugs significantly reduced numbers of all life stages except eggs. And spine soldier bugs consume every life stage the fastest, and nymphs of this thing mass attack their prey. The adults don't have a problem one-on-one, but the nymphs get together in a group and go after them. And the lady beetles and green lacewings did not consume any lanternflies, which surprised me. I thought they would. And this is the last study I'm going to talk about. Since we had a Facebook page, we were getting lots of emails. Hey, I'm finding dead lanternflies at the base of my milkweed. Is milkweed toxic to spotted lanternfly? I don't know. Nobody tested it. So we did test it, and here's a nymph feeding on milkweed. And we compared survival and host preference between common milkweed and grape. And we also did no choice feeding trials and choice tests between milkweed and grape. And when you're doing choice tests, you have to be checking them more often. And we set up cameras as well to help us out. And this is what we found in the no choice experiment. Within 24 hours, every life stage saw a significant mortality on milkweed compared to grape. This is when they only had access to milk. And in 24 hours, they're not dying of starvation. We started doing starvation controls, and it's been studied pretty extensively. How long can they live without food? Because we're thinking about hitchhikers, right? And how long can they make it? It depends on temperature. The hotter the temperature, the less time they can make it. They have a very high resting metabolic rate. And here was when we did a plant choice. And so what you're seeing here is the number of lanternflies. These are insects that are feeding on milkweed. They're in a cage with just milkweed and grape, so they're both present. Or they're on grape, but they're not feeding. That's these bars. And these are feeding on grape, and this is mortality in the black. So even though they don't look like they're feeding on milkweed in here, they fed on it enough that they killed themselves, even when they had grape. This is going to be tested in the field this summer. We have a Northeast SARE grant to look into that. If we plant milkweed around a perimeter of the vineyard, can we reduce the lanternfly population of the adults that come in the summer? And we also tested different species of milkweed. So we looked at, this is swamp milkweed, this is common milkweed, and this is tropical milkweed. All of these are found in the United States. And here's feeding on milkweed only. This is when they had access to grape. So this is how much mortality there was. And you can see that common milkweed seemed to be pretty darn effective. Although with the starvation and the grape controls, the choice experiment wasn't significantly different, and neither were these for tuberosa. So we've been trying to get at the mechanism of this. So we've been measuring carditolides, and we're now starting to also measure pregnant glycosides, which can also be a defense in milkweed. So that's what I'm going to share with you today. I hope you don't get spotted lanternfly, but I expect that you will. You do have to wreak heaven in your state. And we have lots of resources online. We just updated our spotted lanternfly management guide, which is, you can see, contains lots of information, including how to manage it, whether you're a homeowner or you're a vineyard or whatever. And then I would, of course, like to thank all of the people who did all this work. And this is the crew in the Philly area. That they were collecting lanternflies. So we use these big pop-up cages, throw branches in there, and then throw the insects in. They need branches or they get upset. So, did anybody have any questions? Over here.
I have a question about the milkweed. They're very cool. It's super interesting. So with the milkweed, do you think the latex has anything to do with it? Because I can't remember exactly, but tuberosa might not have latex in it.
It doesn't.
Yeah.
Wait, maybe it has it doesn't have cardendula.
Okay.
That is it's the only milkweed species I know of.
So you think it's cardinalized and not the?
It appears that it could be, yeah. We're still trying to, we've got to do a regression type analysis, mortality versus level.
Yeah.
Yeah.
So even though they were feeding on milkweed, could you detect any aversion to feeding on milkweed? In any of them?
They didn't seem to mind it,
Okay.
which was surprising. But I had seen it in the field. I had seen lanternflies feeding on milkweed, and I was pretty surprised. They even did on poison ivy. Yeah.
Staying on milkweed, everybody likes milkweed. So is there a genome for spotted lanternfly yet? There must be.
The genome's been sequenced, and my former grad student, Scott Guy's lab in Hawaii, he's in Hilo, is working on it, annotating it.
So,
It
yeah.
hasn't been annotated yet.
Okay, because there's a pretty canonical set of adaptations that milkweed feeding insects have, right, for ATPases and stuff that you could probably
Yeah.
look at and see, do these things look like they belong?
Right. Well, that's part of the reason why we're collaborating with Sarah Berman and Anurag Agrawal for now, because of all the work they've done on milkweed, and they've done a ton. They're actually running some experiments too, so we're sharing stories. Yeah.
So like the whole situation, it looks like the tree of heaven is the bad child here. So like in Florida's perspective, and the grape growing counties, do you think if we do some sort of eradication program for the tree of heaven, just for the grape growing region, will it going to be helpful to make this bug not get established, at least at the grape growing regions or?
That's an excellent question, and it is being done in Erie County.
Okay.
So in Erie County, Flora Acevedo is the grape entomologist there from our department. And she is looking into that very thing. So they've been getting growers to cut down, they do what's called hatch and squirt. So they hatch it into the tree, spray herbicide on it. And then the trees die, and then they can cut them down. So they've been doing that in area, anywhere near grape, vineyards, to see if it's gonna help.
Is there any positive results so far?
We haven't seen it in Erie yet. Although eDNA's been collected there. But I don't know if it's really been there, or if it was passing through. But yes, cutting down tree, or getting rid of tree heaven will help, I think. Because when we did that experiment, we found that they took a week longer to develop without it, and 2 weeks longer to develop an egg load. And they laid 7-fold more eggs with access to true heaven than they did without it. So it has a big fitness, even though it's not required. Yeah.
Are there known egg parasitoids? Because when you're seeing like hundreds, like thousands of them on the side of the tree, to me that indicates you need to introduce
Yes.
natural enemies. Egg parasitoids seem like the primary thing I would want.
Anastatus orientalis. Yes, there is one in China. And it was investigated by USDA. And it had too broad of a host range. It would go after caterpillars of endangered Lepidoptera, for example. So they dropped it. But it's a shame
There's
because,
no other one.
not that we're aware of it. And they did introduce that parasitoid though in South Korea. And they were getting good control of it, but they're not as picky about host range.
Right? So in terms of potential for spotted lanternfly to end up in Florida, what is known about the temperature tolerance?
Very good question. It's better than we thought. They can tolerate pretty high temperatures, 30, 35 degrees, they do slow down. But when it cools a little bit down overnight, they start feeding again. So they've been tested at a lot of different temperatures, and constant temperatures don't give you very good answers because that's not real. So Melikina, for example, has been doing ramping of temperatures and also changes in humidity to look at those things. And she used Stockton, California temperatures as one of her models. And they did fine. But once you get up about 35 C, 40 C, that can kill them. If they don't move to a cooler spot, what we see them do when it's 100 degrees Fahrenheit is they go down to the duct and get into little cracks in the bark or down in grass or whatever's down there to not be as hot. And they seem to survive it fine. Cold temperatures is what surprised us the most. You can put them in a -20 and they will, the longer you leave them in there, the more you think they would die. But 2 hours, 4 hours, they came out and walking around within about 10 or 15 minutes and no problem. So it's concerning that they don't seem to have, and in China, they are found at pretty hot temperatures.
And so grapes, that's the.
Grapes are the big problem,
That's the big-
but they also go for hops. They like hops too, so whether you like wine or beer, you're gonna. Yeah,
This is not a buggy question, but, because of our citrus problems that we have here, I'm thinking about, like a nutrient supplement for these grapevines to help them get through this low carb diet.
yeah, I don't know about that,
Uh,
because I don't work on grapes, I work on trees,
Mm.
but my colleagues do,
Mm.
and I can ask them about that. There's a woman named Michaela Centenari who's in the plant science department, and she's looking into different types of things that can be done to help grapes get through winter, even when they had some starch loss.
Right, okay.
But I don't know what they're finding so far.
Okay,
They're
that's
also
gonna be-
looking into whether
it's gonna be
pathogens can be vectored. So they're looking at viruses and other types of pathogens to see if they, and so far, it looks like the virus that Blasting sharpshooter transmits that causes
Pearson's disease.
Pierce's disease. Yeah, it looks like lanternflies may be able to transmit it mechanically.
Since this pest is found on so many different hosts, I'm just wondering, have you guys looked at what is the primary driver of their movement? Is it the nutritional needs? Is it the phenology? What is it?
Probably both, but that's a very hard thing to study. So the assumptions have been that when you see a lot of tree heaven, and the trees are starting to really suffer, there's a lot of branch dieback. That's when they're taken off. More than likely because their resources aren't as good. And this time of year is when they're doing mass flights. So they're leaving where they were and moving to new food.
Do you think it could be the volatiles that are driving it? Because of the changes in the plants?
The what, I'm sorry.
The plant volatiles?
Oh, Well, a whole lot of people have studied plant volatile to see if they can come up with a good lure to trap it. And so far, there hasn't been anything that works except in a lab, but it doesn't work in the field. So, yeah, I don't know. But they must be able to detect great venom because they find it sometimes in the middle of the forest.
Yes, I'm gonna be the last, closing with a question, so. So like, I said she was gonna be the last, but then I just discovered. So, I wanted to ask you, if your lab has been, successful with using input pathogenetic method? No, I just did a quick search and I saw that you guys are using the Cyanema species, and I don't know if you've had any success with that.
There are some folks that are looking into nematodes, for control, and I don't know that it's been very successful. They just don't seem to have very many pathogens.
Thank you.
You're welcome.